Hey everybody, welcome back to General Biochemistry Lecture. We are in Chapter 16, which is on the citric acid cycle. This is very much related to Chapter 14, when we covered glycolysis, gluconeogenesis, and the pentose phosphate pathway. It is more directly related to glycolysis. After pyruvate is formed from glycolysis, one of the ways that it can be consumed is through the citric acid cycle. And this is another way that the body can create a lot more ATP and some other energy conserving molecules to eventually make even more ATP down the road. As with all of these cycles, there's a lot of regulation that goes on, and these cycles are not happening in a vacuum. They're all happening at the same time, and they're talking to each other and either turning one on, turning one off, that kind of thing. So we'll talk about all of that in this chapter. First, we're going to start with a little bit of an overview about cellular respiration. So I'm sure that you've heard about this in biology class, but we're just going to review it so that we're all on the same page. Cellular respiration is the process that's used to take pyruvate that's made from glycolysis and further oxidize it to water and carbon dioxide. There are different stages of cellular respiration. So the first stage, we have the oxidation of all different types of fuels to make acetyl-CoA. So we talked about glycolysis and how we can make pyruvate from that. But there are also ways that you can go from amino acids or fatty acids directly to acetyl-CoA. And all of those ways feed into the citric acid cycle. So that's the second stage of cellular respiration. You take acetyl-CoA, you stick it onto an oxaloacetate molecule to make citrate, hence the citric acid cycle. It's also called the tricarboxylic acid cycle, or the TCA cycle, or the Krebs cycle. Krebs is just the last name of the person who discovered this cycle and did a lot of work identifying enzymes and things of that nature. This pathway is pretty much universal, and you generate lots of energy-conserving molecules like NADH, FADH2, and you'll also generate GTP or ATP. We'll get to that. But GTP and ATP are seen as kind of synonymous in the cell in this capacity. The third stage, which we're going to cover in the next chapter, so that's oxidative phosphorylation is what we're going to cover next. Stage 3, we're talking about the electron transfer chain, oxidative phosphorylation, and this is where we generate a ton of ATP. So the ATP that's generated in glycolysis is kind of like child's play in comparison to what you make in the citric acid cycle. And then when you consider oxidative phosphorylation, that makes everything else look like, pennies, drop in the bucket in comparison to the amount of ATP that you make in this process. Here are the key principles for this chapter. As always, look over them, make sure that you understand all of these principles and that you can kind of talk through them a little bit, relate to the topics that were covered in the lecture. So let's get started. We're going to talk about the production of acetyl-CoA from pyruvate. We're going to introduce a couple of cofactors here. So far, we've been talking a lot about ATP and ADP. We had a little bit of a showcase of biotin, but now we're going to introduce a few more players. Coenzyme A is one that you're definitely going to see a lot when it comes to fatty acid catabolism. And coenzyme A has a reactive thiol group that is really critical in its role as an acyl carrier. So acyl just means that it's something that's got a carbonyl on it. So it could be something as small as an acetyl group. It could be something as big as a fatty acid. The thiol group forms a thioester with acetate in acetyl-CoA. So you see this huge molecule. I'm going to switch to my pointer here. This huge molecule is CoA, the entire thing. The business end is here, the reactive thiol group. Seems a little bit wasteful, but This is actually a really good leaving group. So you can leave behind, or you can cleave off the CoA and you can do some kind of other chemistry, just like you use ATP and you phosphorylate something, and ADP is a really good leaving group. CoA is a great leaving group. So there's something called the mitochondrial pyruvate carrier. Remember that glycolysis occurs in the cytosol, and so you make pyruvate, but all of this is happening, the citric acid cycle is happening inside of the mitochondria. So we have to get the pyruvate to the mitochondria where it can then be converted to acetyl-CoA, which can then be shuttled into the citric acid cycle, amongst other things. The mitochondrial pyruvate carrier is a hydrogen-coupled pyruvate-specific symporter, which symporter just means that it carries two items through a channel or a transporter or something like that at the same time. So it carries a hydrogen and it carries pyruvate, and it pumps those things into the inner mitochondrial membrane. I say pump. I don't want to say like it's active like that, but the pyruvate and hydrogen go along for the same ride to get into the inner mitochondrial membrane. Once they pass through that membrane and they get into the mitochondrial matrix, then that pyruvate can be taken up by the pyruvate dehydrogenase complex, or PDH. And this is a cluster of enzymes and cofactors that oxidizes pyruvate in the mitochondrial matrix to form acetyl-CoA and carbon dioxide. What's really cool about this complex is that all of these enzymes are in one big complex, and there's a kind of a swinging arm that brings the intermediates, between enzymes from enzyme 1 to 2 to 3. And there's a lot of regulation that happens here. So we're going to spend a little bit of time talking about this complex. Now, what reaction does it do? The whole reaction that is catalyzed by PDH is the oxidative decarboxylation of pyruvate. It is irreversible, and you remove a carbon dioxide. So it's a carboxyl group at the end that it leaves as a carbon dioxide, and you're left with acetyl-CoA. And there's some stuff that happens in between to make that a reality. Note all of the different cofactors that are required. You need CoA, you need NAD, TPP, lipoate, which we haven't talked about yet, FAD. All of these things are necessary. When I say this complex is massive, I mean it is massive. So here are the enzymes that are involved. We've got pyruvate dehydrogenase, which is E1, and that's the one that uses TPP. And we've seen this reaction before, decarboxylation reaction. You should be able to draw that. Then we have E2, which is the dihydrolipoyl transacetylase. And then we have the third enzyme, which is another dehydrogenase. But this one actually acts on that lipoate arm to make sure that it is reoxidized and ready for more chemistry. So you can see the list of all the coenzymes. This is a beast. There are a couple of other things that are part of this complex, but they're not involved in the chemistry directly. We're going to talk about that when we talk about the regulation of the PDH complex. Now I'll introduce another coenzyme, and that's lipoate. It's got two thiol groups that can undergo reversible oxidation, so they can form a disulfide bond. Think about two cysteines in a protein, how they can form a disulfide bond. It's the same thing with this coenzyme. So the oxidized form is here, and the reduced form is here, where you have two thiol groups. This is attached to a lysine residue, that's a part of the E2 enzyme. So you can imagine if that lysine residue were mutated to something else, that you would completely lose functionality of PDH. So this complex contains multiple copies of E1, E2, and E3. And I'm not going to say the long names of each of these enzymes. I'm just going to stick with E1, E2, and E3. You'll see this complex all over the place in all the species, okay? There may be differences in how many copies of each one. There may be some subtle differences in, the amino acid composition, but by and large, they all do the same thing when you're looking at different species. And you can identify them by sequence, so they're very, very closely related. At the core of this complex, you've got E2. So the one that has the lipoyl arm. And then around that, you've got a bunch of copies of E1 and E3. The structure of the PDH complex is really similar to other enzymes that catalyze oxidations. So we're going to talk about alpha-ketoglutarate dehydrogenase. That is a complex that's within the citric acid cycle. So stay tuned for that one. The branched chain alpha-keto acid dehydrogenase, we're not going to talk about that one in this lecture. In a given species, the E3 complex, or the E3 enzyme, is the same in all three of these complexes, PDH and both of the dehydrogenase complexes that I just mentioned. And their similarities definitely reflect a common evolutionary origin, and they're paralogs. So this is a simplification of what goes on in the PDH complex. You've got pyruvate that has just come into the mitochondrial matrix via that carrier protein, and it encounters PDH. With E1, it's going to be decarboxylated using our old friend TPP. You should be able to draw this mechanism. So you should be able to draw it. It looks very similar to a reaction that occurs in glycolysis. So check back to chapter 14 if you need help with that. But you should absolutely be able to draw the whole reaction mechanism for pyruvate losing that carbon dioxide. You'll be given the structure of TPP, just kind of the business portion. I'm not going to force you to memorize that, but you will need to know the reaction. After you do that, you now have hydroxyethyl TPP. And that interacts with that lipoyl arm, so the lipoate from the E2. Remember, the E2 is kind of like that big core. You transfer this hydroxyethyl group to the lipoate, and in doing so, you create the acetyl portion of acetyl-CoA. So you oxidize that carbon further to make the carbanyl. Then there's a reaction with CoA where you exchange this sulfur for the sulfur that's in CoA. And you're left with a reduced lipoate, right? So the lipoyl lysine is what you'll also see that called, because it's attached to a lysine. Well, we've already made our product, acetyl-CoA, but this enzyme is not restored. We can't do another round of chemistry because the lipoyl lysine is fully reduced. And in order for it to accept another hydroxyethyl group, it needs to be restored. And that's where E3 comes in. It's a dehydrogenase, and it uses FAD and it will accept hydrogens and electrons so that you can reform that oxidized lipoyl lysine. In the process, those electrons are then transferred to NAD to make NADH. So a lot's going on here, a whole lot. That means that there's a lot of things that need to be regulated. Pyruvate dehydrogenase, we said that's the E1 with TPP, catalyzes that decarboxylation. It is the rate limiting step. So the rest of the process only goes as fast as E1. Think about what that means in terms of chapter 6 when we were kind of assigning variables. So think back to your kinetics to kind of remind yourself of what that means and kind of what rate limiting step means. The second step, you oxidize that hydroethyl derivative to make the acetyl group. And then there's what's called the esterification to add it onto the CoA. And then we said the E3 regenerates that lipoyl lysine group. So if you're a word person, these last few slides kind of detail what I was saying verbally with the figure. So both of them are there. Hopefully that covers everybody. We all have different learning styles. So why is PDH complex so cool? It's really cool because it gives a great example of substrate channeling. Once that pyruvate is bound and you make that TPP intermediate, it doesn't leave again until it is acetyl-CoA. So all of the intermediates are passed from one enzyme directly to another without being released. That means that there's a strong drive. Once that pyruvate is bound, it isn't going anywhere, and it is completing the transformation to acetyl-CoA. That long lipoyl arm in the E2, it channels substrate from the active site of E1 to its own active site in E2. You release that acetyl-CoA, and then the arm goes to E3 to be restored. There are tethers to the intermediates, which keeps it in the enzyme complex. That's just so cool. And that increases the efficiency of the overall reaction because you don't have to worry about, well, I hope that this, two little carbon group makes it on over to E2 via diffusion without being snapped up by another process or doing some other chemistry. So you minimize the side reactions that can go on. Now let's talk about the reactions of the citric acid cycle, since we just made acetyl-CoA. This summarizes the entire cycle, and it gives you the names of all the chemistry that goes on at each step. I'm not going to go through and say every single, reaction. You can read this for yourself. But you'll definitely want to take note of the types of chemistry. I will go through and point out the different mechanisms that you'll need to be able to draw. So those will come in the coming slides. But this is a very good review slide to help you think through the chemical logic of what's going on with the citric acid cycle. A key thing to note is that the oxaloacetate molecule that is regenerated each time is essentially like an enzyme. It doesn't actually get destroyed. So you can theoretically oxidize an infinite number of acetyl groups being joined to create citrate and go through the whole path. Throughout the citric acid cycle, at least the first half of it or so, we're making some energy conserving molecules. So we're making NADH and FADH2. You'll need to know where all of the points are that we're making energy. So know which steps. So let's talk a little bit more about the citric acid cycle. In the mitochondria, there are a lot of enzymes. lots and lots of enzymes. And those enzymes and coenzymes and all the things needed for citric acid cycle, electron transfer, oxidative phosphorylation, also oxidizing fatty acids and amino acids to acetyl-CoA, and degrading some amino acids to citric acid cycle intermediates. All of those things produce energy. So the mitochondria is really like the powerhouse of the cell. The majority of the energy yielding oxidative reactions happen in the mitochondria. The complete oxidation of acetyl-CoA to carbon dioxide extracts the most potential energy. But we can't just directly take acetyl-CoA and then just convert it to carbon dioxide, because you actually wouldn't get two molecules of carbon dioxide. You'd get carbon dioxide and you'd get methane. And we can't do anything with methane because most organisms can't actually oxidize it. There are a few bacteria that can, but outside of that, nobody's using methane to do anything. So the citric acid cycle takes acetyl-CoA and adds it onto oxaloacetate, so that now you have a couple of extra carbons that you can burn, make some carbon dioxide, and you're making all of these energy conserving molecules along the way. And you can just keep adding acetyl-CoA to the same oxaloacetate molecule and just crank through the cycle a bunch of times. That way you're getting all of the energy out, and you're not making something like methane that you can't really use. The other thing about the citric acid cycle is that carbonyl groups are more chemically reactive than methylene or methane groups. So if you think about the structure of acetyl-CoA, we've got a carbonyl group that's added, and we can kind of add the methyl group onto the oxaloacetate and make it a methylene group to make it more reactive. And then each step of the cycle, we're adding some kind of energy conserving oxidation, or we're replacing functional groups to facilitate that oxidation. So each step is very much calculated to release energy and be favorable. The citrate that's formed from acetyl-CoA and oxaloacetate is oxidized to make carbon dioxide, NADH, FADH2, and either GTP or ATP. We're going to cover that part when we cover all the different steps and all the different enzymes involved. Now we're starting at the top. We've got acetyl-CoA. It is coming in and merging with oxaloacetate. The enzyme that's needed for this is citrate synthase. It catalyzes the condensation of acetyl-CoA with oxaloacetate. There's a really large negative delta G, and we need that for this because the concentration of oxaloacetate is normally pretty low. And the reason you can get away with that in this cycle is that oxaloacetate is pretty much immediately swept away. So it's used up pretty much right when it's made. So that kind of drives this reaction forward too. Here's the structure of citrate synthase. It's actually made up of, it is a homodimer. And each one of these monomers, which is pictured here, has a really rigid group, a rigid subunit, and a more flexible subunit. Once oxaloacetate binds, it causes a change in the enzyme conformation that creates an additional binding site for acetyl-CoA. So oxaloacetate binding is an example of induced fit. Once that acetyl-CoA binds, then you can have chemistry. And there's another conformational change that allows for chemistry to happen. So it enhances catalysis. The induced fit decreases the likelihood of premature cleavage of that thioester bond that's on the acetyl-CoA. Remember, that is a really easy bond to cleave, and we don't want it to happen before we're ready. Here's the mechanism, and this is one that you'll need to know. And when I say know it, I mean be able to draw this out. I will always give you the structures of the substrates. Like, I'm not going to make you remember oxaloacetate or acetyl-CoA. You're not going to have to remember that whole long arm of CoA. But you will need to know the chemistry that happens. So here we've got a few important residues. His 274, aspartate 375, and another histidine, his 320. We're starting from the point where we have both substrates bound. What's going to happen here is we're going to abstract a proton from the methyl group here. And that is facilitated by the aspartate. Those electrons get shuffled here to create a double bond, and that's the enol intermediate. And then electrons here and the double bond in the carbonyl reaches out and grabs a hydrogen from histidine 274. Now that we've got this enol intermediate, and notice how the electrons have pushed back up onto the nitrogen that's in that imidazole ring of the histidine, that lone pair is important. So when you're drawing this mechanism, make sure that you show the lone pair that's formed. Now we've got this enol here, and that lone pair on his 274 is going to say, you know, I actually really liked that hydrogen. I'd prefer to take it back. So it takes that hydrogen back. You reform the carbonyl. And then the electrons in this double bond jump out to attack the carbonyl carbon and oxaloacetate. And electrons in that carbonyl will jump out and grab another hydrogen from that neighboring histidine, his 320. And you once again put a lone pair of electrons on that nitrogen in the imidazole ring. Once that happens, we need to do a little bit of rearrangement, because we need to actually regenerate that histidine, right? And we need to release some kind of a citrate molecule, because right now what we have is citryl-CoA. That's not good enough. So we need to get rid of this CoA. The way that we do that is with water. So we've got water coming in, and it hydrolyzes the bond here. So you have water attacking that carbonyl carbon, and the result is forming a carboxylate here. And what's released is citrate. I'm not going to go through the details of drawing the hydrolysis. That's something that you should be able to do. but you have the water attacking, and there you go. Think about the histidine, the aspartate, and so histidine 274 and 320, and the aspartate. Think about whether or not they're acting as a general acid or a general base. So if it is donating an electron or a proton, or whether it is accepting a proton. When you read the book, you'll see that terminology about general acid, general base. So make sure that you're familiar with that. That's from chapter 6. The next step, once we've formed citrate, is to do a little bit of rearranging. And we use an enzyme called aconitase, which the formal name is aconitate hydratase, but pretty much everybody will call it aconitase. And this catalyzes a reversible reaction where you transform citrate to isocitrate through an intermediate that's called cis-aconitate. This enzyme is very stereospecific, and it will add water to cis-aconitate, and that is why it is so, you're making something that is specific, cis-aconitate versus trans. So when you add water and you form that intermediate, you form cis-aconitate, and then you lose water and you rearrange. There's a very low concentration of isocitrate, and that pulls the reaction forward. So you'll kind of notice that trend. The oxaloacetate concentration is low, and so that pulls the reaction forward to make oxaloacetate as kind of the last step of the citric acid cycle. So you'll see it kind of come around full circle. Now, aconitase uses an iron-sulfur center. You'll also see that called an iron-sulfur cluster. So if I use that term, it's the same thing. And this iron-sulfur cluster is really important for binding the substrate, which is citrate, and to catalytically add or remove that water. So without that iron-sulfur cluster, you cannot do this chemistry. But there is kind of a second life for aconitase. So aconitase actually exists as two isozymes. You've got the mitochondrial isozyme, which is shown here. It does the chemistry in the mitochondria. It's a part of the citric acid cycle. You also have cytosolic aconitase. That's the cytosolic and isozyme. It does the same reaction, but it also moonlights as another protein in its off time. So if you lose that iron-sulfur center, then you'll get something called, it's an iron response element. Okay. We're not ready for that yet. So we're going to stay on this slide. Thought I had another slide talking about it, but we'll talk about it here. So this is not a major point in terms of understanding the citric acid cycle, but it shows you the diversity of what proteins can do and how they may have multiple functions. So it's kind of like you're getting two jobs out of the same enzyme. So it can function as an indicator of low iron levels. So if you have low iron, then aconitase is going to lose its iron-sulfur center. And this is the cytosolic one we're talking about. Once it does that, it can actually go on and bind to these elements on mRNA that regulate the translation of mRNA to different proteins. And there's a really nice figure in your book that talks about this. So I'm not going to recreate it here, but definitely check that out if it's interesting to you. But essentially what it does is twofold. It stops the translation of ferritin, which is in iron storage, and it promotes the translation of transferritin, which is involved in mobilizing iron. So it will bring iron out. So you stop storing iron, you bring iron out. Once iron levels have been restored, then you'll have rebinding of that iron sulfur center, and you'll have your cytosolic aconitase activity restored. So that's kind of a brief aside. It's not central to the citric acid cycle, which is what we're talking about here. And it's not even, essential to understanding metabolism. But I think that it's a really good example of how efficient our bodies are in terms of being able to use proteins for multiple different metabolic pathways. If you think about iron metabolism as one pathway and the citric acid cycle as another. So now we've got isocitrate, and we have to take that and we're going to form alpha-ketoglutarate. We're going to see alpha-ketoglutarate again when we are talking about amino acids, so get used to it. We're going to use an enzyme called isocitrate dehydrogenase, which catalyzes the oxidative decarboxylation of isocitrate. So to recap the citric acid cycle so far, we have added on acetyl-CoA to oxaloacetate to make citrate. Then we rearrange. Now we're going to lose a carbon. We're going to see that whole thing again. Rearrange, lose a carbon, before we get back to the buildup phase to make oxaloacetate again. This enzyme uses manganese, which is not super common, but you do see it. And that manganese is involved in interacting with the carbonyl group of the oxalosuccinate, and it stabilizes the intermediate that's formed. There are specific isozymes of isocitrate dehydrogenase. Some will use NADP and some will use NAD. And the only reason is because you need to generate both NADH and NADPH. The NADPH is used in anabolic processes, so you're building up amino acids and fatty acids and stuff like that. And then NADH is used in other pathways. So what happens here in a nutshell is you're going to lose the carbon that's highlighted here. You're going to lose that carbon and the oxygens associated with it. Hence, it's called decarboxylation. We require NAD or NADP for this, and you're going to pick up some electrons along the way. You release that carbon dioxide, and then the manganese helps to stabilize the charge and facilitate the leaving of that carbon dioxide, and it stabilizes this intermediate. You don't need to know this mechanism. We didn't really touch any of the mechanisms that involve using NAD or NADP, so don't worry about it. Just know the overall what happens and the fact that manganese ions are required for this to stabilize the intermediates that are formed. So now that we have released another carbon, we are going to release another carbon before we rearrange. So alpha-ketoglutarate dehydrogenase complex, this is another one of those complexes that has E1, E2, E3. and you have substrate channeling so that you don't lose the intermediate. We are catalyzing the oxidative decarboxylation of alpha-ketoglutarate to succinyl-CoA and carbon dioxide. And where we're conserving our energy here is in that thioester bond. So the succinyl-CoA, this right here is conserving the energy in this molecule. This complex is quite similar to the pyruvate dehydrogenase complex that we talked about at the very beginning. Uses the same 5 cofactors, E1, E2, E3, that are homologous. So this is an example of gene duplication and divergent evolution. So they do different, chemistry with different substrates, but the heart of the chemistry is the same. So one complex is specific for pyruvate, another one alpha-ketoglutarate, and then what we have not talked about is taking the branch amino acids like leucine and valine, isoleucine, and you can create different, you're pretty much breaking down the amino acids this way. So all of them have a very similar complex composition. Conversion of succinyl-CoA, which we just made, to succinate. It uses succinyl-CoA synthetase, which catalyzes the breakage of that thioester bond to form succinate. And in the process, we are making GTP or ATP. So there are different isozymes of succinyl-CoA synthetase that will either use GDP or ADP for substrate level phosphorylation. You do need to know that substrate level phosphorylation occurs in the citric acid cycle, and you need to know where. And this is where. When you're taking that CoA off of the succinyl-CoA to create succinate, and this is the beginning of the kind of anabolic half of the citric acid cycle, where we're going to start building up this molecule to recreate oxaloacetate again. This molecule is also a part of the, so when we're counting up all of the ATPs that we're making from the complete oxidation of a molecule of glucose, this is one of the ones that's counted. So when we kind of talk about that towards the end, how many ATPs do you make from the complete oxidation of one molecule of glucose? Don't forget to count this energy. So whether it's GTP or ATP, we count it all as ATP, because you can actually have conversion of the GTP to ATP later on. So here's the reaction. You actually require a phosphorylated histidine residue in the active site. And once you have that, the phosphoryl group is added to ADP or GDP to form ATP or GTP. And like I said, there are specific isozymes for ADP and GDP, and GTP, if that is formed, can be converted to ATP later on. The way that it is converted is by nucleoside diphosphate kinase, which we already kind of touched on in talking about metabolism in general, the introduction to metabolism in chapter 13. So the net result of this activity of this enzyme, the succinyl-CoA synthetase, is creating ATP. Now that we've got succinate, we're going to make fumarate. So this is the reversible oxidation of succinate. It uses a flavoprotein FAD, and you form FADH2 in the process. It's actually a protein that's integrated in the mitochondrial inner membrane. So this enzyme actually moonlights as a part of the oxidative phosphorylation and electron chain craziness that goes on in the mitochondria as well. So that FADH2 that is generated can donate its electrons to the electron transport chain. This enzyme also contains 3 iron sulfur clusters as well as that FAD. The iron sulfur clusters help with transferring electrons, and FAD is one of the cofactors or coenzymes that we talked about in chapter 13 that is usually seen as a covalently attached moiety on an enzyme. We're not going to talk about how fumarate is formed. I'm not going to go into a mechanism here, but you do need to know, in general, it's reversible and it's an oxidation reaction. We can't talk about the citric acid cycle without talking about inhibitors. So malonate is a really strong competitive inhibitor of succinate dehydrogenase, and it's an analog of succinate. So normally you will not see this in cells. But if you add this compound to mitochondria in vitro, which just means, on the bench. So let's say you have some cells or something that you add this to, then you can actually block citric acid cycle activity, which, as you could imagine, would be totally terrible if it were an organism like a living person or animal or something like that. Oh, one more thing to mention. So this is actually a drug that I think is going through clinical trials like right now. Like it started a couple of years ago, and those things take a while. So this is being looked at as a potential chemotherapy drug. So something that can help cancer patients. Now we have the hydration of fumarate to malate. So we're almost done with the citric acid cycle. We generate L-malate. So this is fumarase is a very stereospecific enzyme. So we make L-malate. And the transition state uses a carbocation. Again, we're not going to go through the mechanism here. You're not required to know this mechanism, but know that it is a reversible hydration, and it uses a carbanion to accomplish this. Now that we've got malate, we've got L-malate dehydrogenase, which again, notice it says L-malate, so stereospecificity is key here. And it catalyzes the oxidation of malate to oxaloacetate. So notice how the difference is we've got this alcohol here, and instead, after this enzyme does its job, we have a carbonyl, and we've restored that oxaloacetate. In addition, we made another molecule of NADH. The really low concentration of oxaloacetate is what helps to pull this reaction forward. Without it, the equilibrium would lie more to the left, with just hanging on to that malate. But because the concentration of oxaloacetate is so low, it drives the reaction to the right to make oxaloacetate. And this is how you regenerate oxaloacetate for citrate synthesis. So this is the end of the cycle, and then once you start again, you would add another acetyl-CoA to this oxaloacetate and go around again. This summary figure will definitely help you because you can kind of blank out all of the places where there's production of these energy molecules and refill it in. You can also write down the different reactions that are going on. So like aldol condensation here, this is a rearrangement of some kind, and then you've got decarboxylation. And so you can fill in a lot of detail here on this slide. So I would highly recommend using this as part of your studying for this chapter. This also shows you the different steps where you make all of these energy conserving molecules, the NADH, the ATP, FADH2. So the citric acid cycle directly generates only one ATP per turn. Directly. But because we made all those molecules of NADH and FADH2, all those electrons can be transferred using the electron transport chain to the ATP pump, that the ATP synthetase pump that is active in oxidative phosphorylation. And we're going to talk about that next. Not in this lecture video, but that's the next one to come. So each NADH that you create goes to making somewhere in the ballpark of 2.5 ATP, which I know you're like, 2.5? How do you make half an ATP? It's an average. Remember that NADH can be used for a bunch of other things too. So it doesn't have to just go through this whole pathway to make ATP. It can do other things. Same thing with FADH2. And FADH2 only makes about 1.5 ATPs. So think about all of the molecules of NADH that we made in glycolysis and all the ATPs that we made, plus the ATP from the citric acid cycle and all of our energy conserving NADHs and FADH2s. When you add all that stuff up, you get somewhere in the ballpark of 30 to 32 ATP molecules formed. We haven't even considered the oxidative phosphorylation that occurs in the mitochondria as well. But what I do want you to know is the maximum number of ATPs per molecule of glucose. And so far, our tally is about 32. So you should be able to do the math, not just memorize, okay, 32, bet, but know all of the steps in glycolysis and the citric acid cycle that create ATP directly, or the molecules that can donate electrons to generate more ATP, and how those fit into it as well. Now we're going to talk about how the citric acid cycle actually feeds a lot of other cycles. So it's kind of seen as the hub. The citric acid cycle will accept 3, 4, and 5 carbon skeletons. So some of these skeletons can come from amino acids. We're breaking down amino acids. So for example, deamination of aspartate can give you oxaloacetate. If you deaminate glutamate, you get alpha-ketoglutarate. So we'll talk more about this when we talk about amino acid metabolism later on in the course. So you don't need to know these two examples specifically, but just know that the citric acid cycle accepts carbon skeletons, and one of those sources is amino acids. The citric acid cycle serves as both a catabolic and an anabolic process. So it's called an amphibolic pathway. If you think about amphi as meaning both, so it can do both. Animals can't actually convert acetate or acetyl-CoA to glucose. There are other organisms that can. We're not even going to talk about that because you already have enough information in this presentation. So if you're interested in that, you can look it up. It's in your book, but we're not going to talk about it. So here's the breakdown of how the citric acid cycle fits into anabolism, so building up all these molecules. So citrate can actually be used to make fatty acids and sterols. You can use alpha-ketoglutarate to make, let me switch to my pointer. You can use alpha-ketoglutarate to make glutamate, which can then be used to either make more amino acids, or you can make purines. So that ties into nucleic acid synthesis, right? Succinyl-CoA, we're talking about porphyrins and heme. Pyruvate can actually be converted to malate, which, remember, we talked about that a little bit when we talked about how pyruvate can be converted to, how we can use lactate to convert to pyruvate and some other things, and how we can go backwards. One of the ways that we go backwards, we make malate, which can then get sucked into the citric acid cycle. Oxaloacetate can be used, again, to make PEP, which can then be made into glucose. That's gluconeogenesis. Or you can use PEP to make a bunch of other amino acids. So you don't need to know exactly which, so citrate makes this and alpha ketoglutarate makes this. You don't need to know that, but you need to understand in general that the intermediates in this cycle can feed a bunch of different pathways. We got to talk about another enzyme. I know we've talked about a ton of enzymes and it's a little bit overwhelming. But hopefully when you sit down with chapters 14, 15, and 16, you can kind of see how they come together, and how they talk to each other and regulate each other. And it'll make a little bit more sense. So pyruvate carboxylase catalyzes the reversible carboxylation of pyruvate by bicarbonate, that's HCO3 minus, to form oxaloacetate. And this is a reaction that kind of replenishes oxaloacetate. Could also replenish pyruvate. It requires ATP, and it is allosterically activated by acetyl-CoA. Let's think about that for a second. If we have a high concentration of acetyl-CoA, then that means that we've got a lot of citric acid cycle going on, right? I'm going to abbreviate TCA. It's another name for the citric acid cycle. If we have a lot of acetyl-CoA, then we don't really need to take pyruvate and make more acetyl-CoA. We really don't need that. We've got a lot of acetyl-CoA, so we should probably stop that. And instead, we should take that pyruvate and do something else. We can make more oxaloacetate, which will then help facilitate the increase in the flux through the citric acid cycle, because now we've got more oxaloacetate to accept all of those acetyl-CoA molecules to go round and round. With pyruvate carboxylase, we need biotin, and that's a vitamin. It's a carrier for one carbon groups, so carbon dioxide essentially. And we saw that in glycolysis already. This time we're going to talk about it in a little bit more detail. There's two steps in this carboxylation reaction, and they occur at different active sites. The arm of biotin is what transfers the carbon dioxide from one place to another. So we've got the first reaction, which is activating that carbon dioxide or the bicarbonate with ATP, and you form a carboxyphosphate intermediate. That gets broken down, and then you end up adding it to biotin. That's here. Then you have that biotin arm swinging around and bringing that carbon dioxide to the second active site. You release that carbon dioxide, and what happens is you take the pyruvate and you add the carbon dioxide to the pyruvate molecule, and you form oxaloacetate. I'm not going to require you to know this mechanism. I feel like you've got enough mechanisms to know. But you should definitely understand the reason that you have to have that biotin arm, that you're moving from one active site to another. And again, we're using a lysine residue here. So this is a covalent modification to a lysine in the active site of pyruvate carboxylase. And without that lysine, you can't have activity. The arm takes the product of one reaction and moves it to the other active site so that you can have the second reaction occurring. So we've seen a couple of different examples of what's called biological tethers. And these tethers allow for flexibility, and it also helps with substrate channeling. So if you have this long arm that can move an intermediate from one active site to another, whether that's within the enzyme itself or to another enzyme in a large complex, you're going to increase efficiency and decrease the amount of side reactions that occur. And you'll also speed up the reaction in terms of not having to wait for diffusion for the product of one enzyme to get to the active site of another. Now let's get to the regulation of the citric acid cycle. Regulation balances the supply of all these key intermediates with the demands of energy production and biosynthetic pathways. We said that citric acid cycle can contribute to both. Regulation occurs at several different points: the pyruvate dehydrogenase complex, citrate synthase, isocitrate dehydrogenase complex, and the alpha-ketoglutarate dehydrogenase complex. So these are all places where regulation occurs. Take a second and think to yourself, what kinds of molecules do you think will activate and which kinds of molecules will inhibit the citric acid cycle? If we're talking about low energy molecules like AMP or ADP, high energy molecules like ATP, acetyl-CoA, how will they affect all of these complexes? You're going to get the answer, but take a second to think about it before you move forward. And this will help in your thinking with all of the metabolic pathways going forward, to think about them in generalities versus memorizing each specific allosteric regulator. So for the pyruvate dehydrogenase complex, activity is turned off when you have a lot of fatty acids and acetyl-CoA. And that makes sense, because if you have a lot of acetyl-CoA, you don't need to make more. And that's what the PDH complex does, is it makes acetyl-CoA. Oftentimes you'll see these big complexes are inhibited by their product. And that is by design, so that you don't keep making the product and investing energy and resources when you already have enough. If you have a lot of fatty acids around, that also indicates that you're doing fatty acid synthesis, which implies that there's a lot of acetyl-CoA around as well. What also turns off this complex is lots of ATP and NADH. So we talk about it in ratios. And when the ratio favors the high energy molecules, you're going to turn off the PDH complex, because the citric acid cycle makes more of these, and we don't need any more. We've got enough energy. We're good to go. The PDH complex is turned on when you need a lot of energy. So think about the molecules that determine that. We're talking about high levels of AMP, ADP. Those are things that will turn on the PDH complex. We're turning on this complex because we want to make more acetyl-CoA so we can meet the energy needs of the cell. So this shows you all the different allosteric regulators and where they regulate. But you can categorize them in two categories: low energy and high energy. And once you categorize those allosteric regulators, it will make a lot more sense, and you'll be more fluid in talking about this and thinking about this. So I highly recommend that you think about these allosteric regulators in groups versus individually. I also want to note that we have calcium added here. Remember that for muscles, release of calcium means that the muscle is contracting, and that is, a state where you need more energy. The muscle is like, hey man, we're doing a lot here, we need some help. And so that signals for all the things that we talked about in chapter 14 and 15. Also of note, high amounts of succinyl-CoA will also inhibit the alpha-ketoglutarate dehydrogenase complex. So it's another example of a product inhibiting the complex that makes it. Succinyl-CoA will also inhibit citrate synthase. Another way that the PDH complex is modified is through phosphorylation and dephosphorylation. So at the beginning, we said that there was more to come in terms of regulation and also what's a part of the PDH complex. So the PDH kinase and the PDH phosphatase are also part of the complex. So two regulators are going along for the ride, making it a lot easier and quicker to very, very quickly modulate the flux through the PDH complex. So the kinase inhibits PDH activity. So when it's phosphorylated, it's inactive. When that phosphoryl group is removed by the phosphatase, that reverses the inhibition. When you think about it, phosphorylation requires energy input. It requires ATP, right? And if you have a lot of ATP, then you've got some extra ATP to be phosphorylating some things. And if you phosphorylate PDH, then that says to it, hey, we've got so much energy around that I can phosphorylate you. So you should be quiet, take a break, take 15, mandated by law. You got to take a 15 every so many hours. Take your break. We'll let you know when to come back. The phosphatase says, oh, hey, we need you back now. So I know you were comfortable, hanging out, listening to music on your phone, scrolling through the gram, whatever. I'm going to take this phosphate group off of you so you can get back to work. Thanks. So we said that regulation occurs at a bunch of different places for the citric acid cycle. We just covered where all the regulation is for the pyruvate dehydrogenase complex, which isn't exactly a part of the citric acid cycle, but it feeds into it. Then we've got these really exergonic reactions that are catalyzed by citrate synthase, isocitrate dehydrogenase complex, and the alpha-ketoglutarate dehydrogenase complex. All three of these are also regulated. So they're affected by the concentrations of their substrates and their products. So ATP and NADH are inhibitory. That makes sense. They're high energy molecules. NAD and ADP, they stimulate activity, and these are low energy molecules. And in addition to that, we've got long chain fatty acids, which are not exactly high energy molecules per se. You can break them down to get energy, but this indicates high levels of acetyl-CoA, which again serves to inhibit. So there are some mutations that affect the PDH complex and citric acid cycle enzymes. And those mutations are oncogenic, meaning that when they happen, it allows for, unchecked cell growth. Some of these mutations are things that downregulate the mitochondrial pyruvate carrier, which means that you have a decrease in the flux of pyruvate into the mitochondria. And that means that there's more pyruvate available to go in reverse. So remember how we can just make lactic acid and then take that, make oxaloacetate, and then go from there to make PEP and then do gluconeogenesis. Cancer cells prefer to use glycolysis and crank through that versus using the citric acid cycle. There's some contemplation that perhaps one of the reasons why this is because if you're doing a lot of cell growth and mitosis, you need a lot of energy, you need to make a lot of things. But if you keep doing that in the mitochondria, you're making a lot of reactive species. And those oxidative species can do a lot of damage. So if you're increasing the flux through the mitochondria, then you could potentially do harm to the cell by creating all these reactive oxygen species. So instead, you do glycolysis, which it's not as efficient. You're not getting as much energy, but you aren't making metabolites that are potentially detrimental to the cell. Other mutations will inactivate the PDH complex, which means that you are not making acetyl-CoA, and instead that pyruvate is being again shuttled through to make lactic acid or something like that. You can also inactivate succinate dehydrogenase, and that would again shut down the citric acid cycle, which is bad. Oncometabolites are ones that stimulate tumor growth by acting through specific G protein coupled receptors in the plasma membrane. So some of the metabolites can go and signal for other things to happen. These signal cascades occur, and they favor tumor growth. Lactate and succinate are two such oncometabolites. So they will stimulate unbridled, unchecked cell growth. Mutations in succinate dehydrogenase and fumarase can cause tumors, which means that they're considered tumor suppressors. And glial cell tumors have this mutant NADPH dependent isocitrate dehydrogenase. And what's wild about this is that in the mutant, you actually can't convert isocitrate to alpha ketoglutarate. You instead make 2-hydroxyglutarate, and then that can go on to do some other things that's beneficial for the cancer cell. For all of this stuff about cancer and the mutations and stuff, you don't necessarily need to know this part. This is not where the focus is. The focus is on knowing the citric acid cycle, but this is definitely a part of kind of the wow factor, or kind of why this is important. Why do you need to understand the different enzymes in the citric acid pathway and how it ticks, how it's regulated, to understand what happens when it breaks and why it's so detrimental when it breaks. So there are some intermediates that are channeled through what's called metabolons. Metabolons are integrated multi-enzyme complexes that are held together by non-covalent interactions. And it is believed that malate dehydrogenase, citrate synthase, and aconitase are part of a metabolon. When you look at cells, intact cells, you'll see these non-covalent interactions, you'll see these enzymes hanging out with each other. But when you try to purify the complex, there's no complex to be had. In the cell extract, you've got something different. You've got, dimers or things like that, where you don't see an intact complex with all of these enzymes included. So there may be some conditions, either it's just the concentration of the proteins is too low, or other things that occur when you break open a cell that cause the interactions to be broken. That's it for chapter 16. Thanks for watching. Hopefully this wasn't as intimidating as chapter 14. I think that one was probably the biggest chapter so far. But I do want you to take the time to relate how these cycles are all related. So think about glycolysis, think about gluconeogenesis, the pentose phosphate pathway to a lesser degree in terms of being related to the citric acid cycle, glycogen breakdown and making glycogen, that's definitely related to glycolysis and gluconeogenesis, and how all of those things play with the citric acid cycle in terms of the regulatory molecules. So all of the allosteric regulators that we've been talking about, they all affect these processes. So if you have an increase in calcium, let's say in the muscle, what's going to happen? You need to think about glycolysis, gluconeogenesis, and all these other things, whether they're going to be turned on or off, and then what the net result is going to be. Are you going to increase energy production? Are you going to increase blood sugar? Are you going to increase, the amount of glucose available to the muscle cell? What is the end goal? So think, kind of small picture, kind of narrow focus in terms of the mechanics, but also big picture, what's going on, and overall, what's going to be the metabolic change, like change in blood sugar or change in energy, big energy production. Those are the tips that I have for you with studying the chapters that we've covered so far in this section. Please take the time to put this information together, make your own figures, talk it through, all that kind of thing, because it will really help you understand how things are regulated. And then practice drawing out the mechanisms. So I'm not going to go through and draw every mechanism each time, especially if we've already covered it and you've had organic. So these are things that you should be able to do. But if you need help, you can always come to me in office hours. All right, I'm going to stop talking and rambling. Thanks again for watching, and stay tuned for the next one. Be safe.